Okay, so this is the first lesson in Upper Sixth, where I'm just going to recap the introduction to genetic control circuits that we're going to come across when studying gene regulation in both bacteria and in eukaryotic cells as well. So gene regulation, hopefully you're aware, is the regulation of turning on gene products or synthesis of gene products at the right time and place, and then turning them off again when we no longer need them. So in our cells, that might be, production of hormones, so production of insulin, for example, a peptide hormone, so it's a protein, and we turn on transcription and then subsequent translation into a protein when insulin is required by our body. In other words, once we've had a high sugar diet, so lots of glucose in our blood. In bacteria, they may use control circuits in a similar fashion to regulate gene expression by turning on genes required for lactose metabolism, for example. And I use that as a discrete example for a reason which you'll see next lesson when we actually look at lactose as a specific example of a genetic controlled circuit. Ultimately, control circuits have the following properties, and this is why they're important. They conserve energy and resources. That is primarily the reason why cells, bacteria in this case, use control circuits. The reason is to conserve energy, to not waste resources. It is achieved through primarily feedback inhibition, which you should have covered last year in lower sixth at least once. The example I give to my divisions is the allosteric inhibition of the enzyme phosphofructokinase, which, as we all know, is an enzyme in one of the stages of glycolysis, and it is inhibited by ATP. So ATP will bind to that enzyme, cause a conformational change by binding to an allosteric site, allowing that enzyme to no longer function. It is inhibited. The production of the substrate by that enzyme is inhibited by ATP. And the whole purpose is to maintain homeostasis within the environment, either intracellularly or exterior to the cell. It is achieved through environmental sensing. So in the case of a bacterium, for example, it can sense its external environment and its internal environment and can turn on, induce, we'll come to that word in a minute, or inhibit various biosynthetic pathways as a result, as a direct result of the environmental composition, depending on what molecules are around in the environment or indeed in the cell itself. All control circuits that we're going to cover, I'm going to give you 4 types in a minute as a sort of general intro. They are what we call or refer to as bistable. This just simply means they act as a light switch, but not a dimmer. So it is all or none, all or nothing induction. So the genetic switch, the circuit in question is either on or off. It is not half on or half off or a quarter on or a quarter off. It is bistable. It is either on or off. And that will become hopefully more obvious to you, and you'll understand how that works later on when we look at operons and specifically the lac operon. So let's look at a typical example, just using a sort of generic process here, where we have a substrate on the left there, the circle, and it is being converted into a product, usually by, if this is one step of reaction, by one enzyme, but typically multiple enzymes are involved here. But for now, just look at this as a one-step reaction. The product, more often than not, will inhibit via feedback inhibition, again, you covered it last year, the synthesis of itself. In other words, when you've made enough product, it will inhibit its own synthesis. In the case of earlier, and certainly my divisions should know this from lower sixth, ATP inhibits the enzyme phosphofructokinase, which is part of the glycolysis, the start of respiration. The whole point of that is respiration produces ATP. That's the whole point. But once you've made enough ATP for as a cell, what you require, you don't need any more of it. It's a waste of energy, looking at that top bullet point there, and it's a waste of resources to continue to make ATP because there's actually a lot of reactions that happen in order to do so. It's just a waste of energy. So once you've made enough of the product, whatever that may be, you nearly always want it to repress, to turn off the synthesis pathway that makes it. And we call that feedback inhibition. It's feeding back into the same biosynthetic pathway, and it is inhibiting its own production. And that is to maintain homeostasis. So keeping the levels of whatever product we're looking at here, this purple box, whatever that may be, keeping the levels at homeostatic levels, whatever they may be. Of course, you could obtain the product from outside of the cell. No need to make it if you have enough of it knocking around in the extracellular environment. So you could import that into the cell, and you can imagine that that is also going to feed into this circuit, and it will also inhibit its own production. The reason you might want to do that is because if you have it available to you, whatever this product is that you're trying to make, if you have it available to you in the local environment, it is relatively, energetically speaking, for, let's say, your energy source, than it is to have to make it yourself from the substrate on the left-hand side there, the brown circle. You could, however, have an alternative source that you can bring in from outside of the cell that might be a better source of energy than the purple boxes over there. So the red triangles there represent an alternative molecule that is perhaps better. It's a preferred energy source than the purple one. It might have more energy liberated when you metabolize it through various catabolic processes. So if that's a much better source of energy and you would prefer to use that, you can hopefully imagine what it's going to do and how it's going to feed into this genetic circuit. It is, of course, going to inhibit the production of our purple product there because that is substandard. There is no need to make that from the substrate if ultimately we would rather use the red triangles there, an alternative source of energy. And the example that we'll come across in future lessons, looking at lactose specifically, lactose is represented by the purple box here. It is a useful energy substrate, but the preferred energy substrate for bacteria is glucose. It is more energy rich. Reasons for that, hopefully you can appreciate from going back to your lower sixth notes on the structure of disaccharides and monosaccharides. So we prefer to use glucose in this case. So glucose, the red triangles, can be imported in, and if there's enough glucose around, we turn off the production of our ability to metabolize lactose. And we'll get to that later on. You could also have alternative molecules in the environment or inside the cell even that do the opposite of all this, that are an environmental signal telling the cell that we need to produce the product, whatever that may be, an enzyme, for example, involved in lactose metabolism. But it could be anything. This is just a generic overview. In terms of names, we have induction here. So if you are turning on a genetic circuit, if we are making product, if you're biosynthetic pathway, we call it induction. You are inducing the formation of product. Feedback inhibition, you have come across before. It is inhibiting the production of the product. And if it is feedback inhibition, that is the product itself feeding back into its own synthesis pathway and inhibiting its production. The third one, which is also new to you, is our preferred substrate, or our preferred molecule that we would rather have in our cell, and we therefore don't need this other one, are the red triangles. That we call catabolite repression. So it will also repress this biosynthetic circuit, but it doesn't have to be the product. It's an alternative molecule, an alternative, in this case, energy source. So how does this work in kind of real life, in your life perhaps? This might represent a drink on a Saturday or Friday night, for example. And too much of that requires you to have energy. So you then go looking for food of some sort. In this case, fish and chips from a chip shop. And that's nice and easy to get hold of. So you could imagine that this therefore inhibits or represses alternative sources of energy. In this case, either microwave chips or do it yourself with a deep fat fryer chips. The reason for that is because it's much easier and quicker to obtain. And why bother making it yourself? Why bother making the chips yourself when it tastes so much better from the chip shop anyway? So you can imagine that that's going to inhibit, and also there'll be no sort of risk of oil fire, deep fat fryer spills and chip pan fires. You could imagine, though, that you could get this food that you desire from alternative sources. In this case, this fan down here. That also could inhibit the alternative source, the do-it-yourself-at-home method. But you could also imagine that the fish and chip shop is still a superior energy source. It's still quicker and probably tastes a lot better. So that you hopefully can appreciate will inhibit also the alternative energy source. Now, there is a problem which I've not mentioned, which is, of course, that all of this leads to a sort of positive feedback loop in both cases, requiring the desire for more drink there, but I think the less said about that, the better. So, before I go into the 4 types of genetic circuits that you need to be aware of for A-level biology, I'm just going to notionally mention these 2 chaps here, Francois Jacob and Jacques Monod, 2 French geneticists who came up with this idea in the 1960s of genetic control circuits through their study of E. coli. And this is a quote from their paper talking about genetic circuits. The paper I have linked in OneNote for my divisions. So, just to recap, 2 types of terminology that I have covered so far. I'll do that in a minute. 4 types of control circuit that we are going to look at are shown here. You can be positively controlled or negatively controlled, and I'll come to that in a minute. And in either case, you can be inducible, you can have an inducible circuit or a repressible circuit. So we have 4 types of control circuit that I'm going to go through in a minute. Right, hopefully this should be the terminology. Yes, it is. So let's look at our diagram again. We have our substrate going to our product, and there are 2 overarching options here to us. We can either induce this process or we can repress it. Repression means to turn off synthesis or turn off this particular pathway. And induction of the pathway means to turn it on. Repression occurs by this triangle in this case, and we call that a repressor. It is a protein, typically a protein, called a repressor, that acts to repress or bring about repression of this genetic circuit. Induction is done by an activator. So in blue here, we call this an activator. Again, it's typically a protein, and it will bring about induction. So that's just one to be careful of. We call it an activator protein rather than an inducer. Inducers do exist. We'll get to them later. But the proteins that directly modify either induction or repression are called activators if they cause induction, or repressors if they cause repression. So the first of our 4 genetic circuits is something known as the negatively controlled inducible circuit. It is negatively controlled. That's the first thing we need to think about. And what this means is, whether it's negative or positively controlled, refers to whether the particular protein controlling the either induction or repression of the circuit is either an activator or a repressor. If it is negatively controlled, I hope you can appreciate we are looking at a repressor. So the repressor, here shown in red, is going to repress this genetic circuit, and that will turn off synthesis of our product. It is, however, inducible, and this is where the sort of latter half of the name becomes important. How can we induce this circuit? How can we turn it on? And, think in terms of sort of computer science here and sort of logic gates and that kind of thing, if you're into that kind of stuff. You hopefully can appreciate if you want to turn this circuit on, you need to get rid of the repressor somehow. We do this by adding, and this is where it gets a little complicated, we add not an activator because that would directly cause induction. We need to get rid of the repressor. So we call this an inducer. So the inducer will inhibit the repressor, typically by binding to it, causing a conformational change, possibly at an allosteric site, making the repressor not able to bind to the either the substrate or whatever it is it binds to. Either way, the inducer inhibits the repressor or represses the repressor, and the repressor can no longer repress the synthesis of the product. So therefore, we then get the product formed again. I've put a star in the top right-hand corner because this is one of the 2 control circuits that you definitely need to remember because they come up in specific examples in future topics. Okay, negatively controlled repressible is the second type. So again, it is negatively controlled. Therefore, we're looking at a repressor as the predominant, the sort of main star of the show, if you like. But this time it's repressible. So let's have a look at the repressor. Again, it is repressing this circuit. However, because it is repressible, in this case, we have something known as an apo-repressor, which is not a different repressor. Apo is just a posh biological term that we use to mean inactive. So all it means is there's a repressor still. It's just not constitutively active. It's not able to function by itself. So the default position for this particular circuit is that we are making product in the first place, as opposed to the negatively controlled inducible circuit we've just seen, where the initial state is, of course, that the repressor is repressing the circuit and the synthesis of the product is turned off. So how are we going to repress this circuit? So we've dealt with the negatively controlled bit where we start with a repressor, but it's repressible. So that means when we add something, a compound of some sort, we cause repression. That's the end state. As opposed to the negatively controlled inducible we looked at, where the end state is induction. So the first half of this, negatively controlled, again, refers to the repressor. being the main star of the show. But then the second half, either repressible or inducible, refers to the overall, the end product. Are we repressing or are we inducing? And in this case, we're going to repress. So how are we going to repress this circuit? Hopefully you can appreciate that we're going to do something to the repressor to make it able to function. And in this case, we add a co-repressor. Again, a protein perhaps that binds to the apo-repressor, alters its conformational shape, and allows it to then bind to whatever it needs to bind to cause repression and therefore turn off the genetic circuit. If you remove the co-repressor, the repressor is no longer repressed, and then of course you can turn this circuit back on again. And apologies, I don't know where the product is. It should have reappeared. Okay, the next two are positively controlled circuits. So, in contrast to the other two, the positively controlled circuit, and actually I'm just going to go back actually to this one, just to confirm that if I get this back up again, here we go. Why is this one repressible and the first one inducible? Just to reiterate the point, it is because when you add both components, you add the repressor, and in this case, the co-repressor, you are causing repression. So that's why it's repressible. In the first example, it is negatively controlled inducible. Why is it inducible? Because when you've added both components, the repressor and the inducer, you are causing induction. We are turning on the genetic circuit, just to make that clear. So, positively controlled inducible, hopefully you can think about this yourself now as to what's going to happen. It's positively controlled, therefore we need to think about the star of the show not being a repressor, but being an inducer, or an activator, actually, at the start. So let's put the activator in. Here we go. And it is inducible. So we must start with an apo activator, an activator that does not function by itself. Because when we add the second component, we want to induce the circuit. It's inducible. So the end state has to be that the switch is turned on. So we have to add a co-activator. That modulates the apo activator to turn it into an activator, which then causes induction and production of our product. If we remove the co-activator, as before, then the apo activator returns to its sort of inactive form, and the genetic circuit is turned off. Again, that is an important example to know, and it will come up in a future lesson. The final type that you should know is the positively controlled repressible. So in this case, we have our activator, and it does function by itself. So this will stimulate this process, it will induce the genetic circuit, leading to induction and production of the product. But the end state is repressible, so we are able to repress this circuit by modulating the activator. And how we do that is we add an inhibitor that will bind to the activator, inhibit it by changing its shape, or causing a conformational change, and that will mean that the activator can no longer bind to its particular ligand or substrate, and therefore it will turn off production of the product. The genetic circuit is turned off or repressed. So, the final thing, as to this sort of introduction to genetic circuits, is just to consider on which level we are going to control gene expression. And which level genes are controlled depends very much on what type of cell you are, whether you are eukaryotic in terms of sort of your kind, your cells, or whether you are prokaryotic, so archaea and bacteria. Again, I sort of make the point that in your exams, they are synonymous terms, but in reality, the word prokaryote is an outdated taxonomical term because archaea are indeed prokaryotes as well. They have no membrane-bound nucleus. So just saying prokaryote doesn't necessarily refer to bacteria. So I will typically say bacteria in this case, but in your exams, very often they will be referring to bacteria. So hopefully we're aware of transcription by now, whereby at the start of the gene, the enzyme RNA polymerase will bind. It knows where to bind because there is a sequence motif at the start of the gene at the 5' end of the coding information called the promoter. And the promoter is the sequence that allows RNA polymerase to bind to it and to unravel the double helix, forming a transcription bubble by breaking some of the hydrogen bonds in the local vicinity. The RNA polymerase can then translocate along one of the strands of DNA, copying it into an RNA product. And in the case of bacteria, because that's what we're going to focus on, the product is mRNA. There is nothing that is needed to be done to the RNA product before it is mature and ready to be translated. That contrasts, of course, with eukaryotic transcription, which we will cover in more detail later on, because there are some more problems associated with that. Either way, off we go, and then the RNA polymerase will, of course, stop transcription when it reaches the terminator sequence. So let's do that there. And the mRNA is produced. Translation is then when we take the mRNA product we've just made, and it is bound by a ribosome, which translocates along the mRNA using GTP hydrolysis. Interestingly, not ATP hydrolysis, though we release free energy from the hydrolysis of the phosphine hydride bonds in GTP. Hopefully you're aware of that from last year. It is a minor difference in that GTP is used here as the substrate rather than ATP. So it's one of the few examples where ATP is not used for energy. So that will translocate from the start codon, which is typically AUG from the 5' to 3' direction. And it will reach one of the 3 stop codons, TAA, TAG, or TGA. In the case of DNA, obviously, U is substituted for T in RNA, so it would be UAG, UGA, or UAA. That happens there, and of course, your amino acids are joined together via a condensation reaction, producing peptide bonds, and your protein, which can then fold up into its various secondary structures and then tertiary structures, and more often than not, it will then associate as a subunit as part of a larger complex, quaternary structure. So I would ask you to have a think about what level you would want to control gene expression at, depending on what organism you are. So pause the video, have a think, and we'll talk about it more as we cover the next lessons. But ultimately, the sort of salient point of the news is that bacteria can couple transcription and translation together. That is because they don't have a nucleus. So the mRNA here, as soon as it is produced from transcription, can be bound by ribosomes to do translation, almost as it is, well, at the same time as it is being synthesized. In the case of eukaryotes, we can't do that. We have to modify the mRNA first, and we then have to export it out of the nucleus, where it can be bound by ribosomes in the cytoplasm. So therefore, bacteria can regulate very easily at the transcriptional level to very quickly turn on transcription of genes, because translation follows pretty quickly afterwards. Humans or eukaryotes, certainly higher eukaryotes, tend to regulate at the post-transcriptional or translational level. More of this in future lessons, but that is because it takes much longer for us to transcribe our gene into RNA, pre-mRNA, to then deal with it and make all of the chemical modifications that we need to, again, more on that later, and then export it into the nucleus and bind it to a ribosome and then carry out translation. We then, of course, might need to modify the proteins in the Golgi apparatus and various other organelles. So in eukaryotes, we regulate predominantly at the level of translation, or actually post-translation, and more on that later. That is not to say that we don't regulate at the level of transcription, but there's a lot of regulation that is carried out at the translational level. And that's because ultimately it takes us longer to produce our proteins. So if we need to quickly, as a cell, produce lots of insulin, for example, or activate membrane transporters, that kind of thing, bacteria would transcribe those genes, make the protein, and then insert it into the membrane. In our case, we would probably rely more often than not on having those proteins available to us and then just regulating post-translationally by altering the protein to turn it on. And that allows us to regulate gene expression quickly.